A new generation of science is emerging from Africa where innovation is engineered and solutions are delivered across education, biotechnology, healthcare, and technology. This is Genomac Holdings, and we are a multidisciplinary innovation company transforming lives through genomics, artificial intelligence, and education. Across our subsidiaries, we are translating research into impact. As Genomac Labs, we advance precision medicine. As Geneious, we harness nature to create sustainable therapies. As Geneious Services and Consults, we empower institutions with innovative scientific services. At Genomac Innovation Hub, we build startups that redefine Africa's health future. And through the Genomac Institute Incorporated, we are driving education and research in genomics and bioinformatics. Today, I introduce GOGeneneBio, an initiative that brings genomics education directly to communities worldwide, equipping people with practical skills to innovate, build, and lead in today's biotech era. From Africa to the world, Genomac Holdings is forging a sustainable future powered by competence, innovation, and collaboration. Welcome to Genomac Holdings.
Hello, everyone. I would like to welcome you to the global outreach on genomics and bioinformatics. And I would also like to thank you for being our host in advancing genomics and bioinformatics education. All right, so in this session, I'm going to be taking you through Introduction to Genomics. As you all know, this particular initiative is actually dedicated and designed for those who are just finding their feet in genomics and bioinformatics. So this initiative is to help you to be able to understand the fundamental or to gain a fundamental knowledge of what genomics and bioinformatics is all about, and also to be exposed to hands-on training on genomics and bioinformatics. So on my outline here, I'm going to be taking you through the definition of genomics, definition of basic concept in genomics, central dogma of molecular biology, which is a very important aspect of molecular biology, and then application of genomics. So let's get started. What is genomics? Genomics is a branch of molecular biology. Yeah, so I know you must have heard of molecular biology before. Maybe you've not heard of genomics before, but now you know that genomics is actually a branch of molecular biology and it studies the entire DNA set of an individual. So now when we talk about an individual here, we are actually talking about you know, human, plant, animals and microorganism. So in that case, we can say human genomics, that is the study of human genome, the entire DNA set of human. We can say plant genomics, the study of the entire DNA set of plant. And we can also say animal genomics, which is the study of the entire DNA of animals. We can as well say microorganism genomics, or we can say microgenomics, or we can say um, pathogen genomics, or we can say bacterial genomics, or viral genomics, or parasite genomics, or parasitic genomics, as the case may be. So this is just basically the study of the entire DNA set of a particular organism. So it studies the structure, the function, and then the evolution of the genetic makeup of such organism. And you should know that evolution is an important part of genomics because this is what helps us to trace the changes that has occurred over time, for a particular defect, in a cancer patient, in a diabetic patient, in a patient with neurodegenerative disorder, as a result of changes that has occurred. Even in a microorganism, you will discover that there are some microorganisms that long before they were not resistant to a certain antibiotics, but over the time they've develop resistance to an antibiotics, and this is as a result of evolution or changes that has occurred in them. And genomics helps us to be able to understand or gain insight to these changes. All right, so now let's define the basic concept in genomics. Now, the word genomics is from the root word genome, which is the organism's complete DNA set. So, just like we have human genomics, which is the study of human genome, we can say human genome, plant genome, that is the DNA of the complete DNA of human, the complete DNA of plants, the complete DNA of animal, the complete DNA of microorganism, could as well be referred to as, human genome, plant genome, animal genome, bacteria genome, So now, when we talk about molecular biology, it is basically, you know, it has to do with the molecule of life. And when you talk about the molecule of life, you're talking about the DNA. Now, you will remember in school, we're taught, the level of organization of life. And it starts with the cell, the tissue, the organ, and the system. Now, genomics help us to unpack the cell down to the nucleus. And you know that within the nucleus, you have the chromosome, and the chromosome, we have the segment of genes, and when you unpack the genes, you have the DNA, and then down to the nucleotide basis, which is the ATCG. Now, the chromosome is the structure found inside the nucleus of a cell, and it is made up of proteins and DNA organized into genes. And the gene is the basic physical and functional unit of hereditary specific sequences of DNA that encode instructions on how and when to make proteins. But this all boils down to a particular material, which is the DNA. Now let's talk about the DNA. The DNA is the deoxyribonucleic acid. It's a double-stranded molecule that stores genetic information. This is where genetic information are stored. Now, every organism, every living thing has a DNA, and the DNA encodes information that dictates life's activity, information that dictates the color of your skin, the color of your eye, the color of your the shape of your nose and other activities, structure and function like that. And then we have the RNA, which is ribonucleic acid, which is a single-stranded molecule, involved in protein synthesis and gene expression. Lastly, we have the nucleotide bases, which is also referred to as nitrogenous bases. They are the language of life, the building blocks of DNA, which is the ATCG. I mean, who could have thought that these simple letters are the ones that encode information that pertains to how you look or how you behave? So these bases form genetic code. Adenine pairs with thymine in DNA, and uracil pairs with cytosine. Oh, sorry, uracil pairs with thymine in RNA, while guanine pairs with C, both in DNA and RNA. All right, so now let's move on to the next thing, which is the central dogma of molecular biology. This explains how the information from the DNA is being translated to protein, because protein are functional elements. Proteins are what are responsible for, your skin color, responsible for the growth of your nails, responsible for um, functions and structures of, of the human biological system. So the DNA itself replicates itself. So think about the DNA as an original copy, or as a material that carries the original copy of genetic information, which and cannot go out, but it has to replicate itself. And when it replicates itself, it transfers this genetic information into the RNA, which is referred to as transcription. And then this information is then passed down to make proteins, which is referred to as transcriptome. So this is just the flow of genetic information. And this flow is actually important in helping us to understand, the molecule of life. Each of these levels, from DNA to RNA to protein could actually be studied to help us understand, you know, um, a particular condition, to help us understand what evolution has taken place, what changes are taking place, you know, which could be the effect of a particular condition like cancer, like a neurodegenerative disorder, or why a particular microorganism is resistant to an antibiotics. All right. So now let's move on to applications of genomics. Genomics and bioinformatics can be applied across various fields. Now, these are just some major ones I've highlighted here. We have various other fields that you can apply genomics. It can be applied in infectious disease, and which are some of the things I've been mentioning lately. When you want to identify the genes that confirs, some antimicrobial resistance. You want to understand how these genes, how they confer resistance to antibiotics and how they go about their resistance. So genomics and bioinformatics help us to be able to identify these genes. And then it's also useful in agriculture, for crop improvement. It helps us to gain insight into plants pathogen interaction. And also, um, let's say a plant is doing well in a particular location, and the plant of the same species is not doing well in another location. The soil composition could be studied. For example, you know that soil, within the soil, we have some microorganisms that are living within the soil, and they could, you know, in one way or the other, improve the crop or the plant that is doing well in that region. So such soil could be studied to understand, the components or the compounds that is actually locked within this microorganism that make this microorganism to, enrich the soil, and as a result make the plants to be you know to be doing well in that region. And such could be studied and then incorporated into this other region or into this other soil or the environment of that plant to improve the plant that is not doing well. Okay, we also have personalized medicine. Personalized medicine is actually a very important aspect in healthcare or in medicine. And, this is what help us to study the genetic makeup of an individual, because we realize that the genetic makeup of everyone is different, because, we all have DNA, but our genetic makeup is different. So the treatment that may work for someone may not work for another person. So genomics help us to study the genetic makeup of an individual. You realize that, for example, a patient who has diabetics but has foot ulcer, could have, you know, could respond to treatment that is given to him or her, why another patient is not responsible responding to a treatment that is given to him or her. In that case, you want to understand, you know, what what is going on, what happened, what is going on, what is happening, because that will help you to be able to, manage the patient better. Maybe it is as a result of, the genetic makeup, or maybe it's as a result of the different strains of bacteria that are present in the foot ulcer, in the wound, you know, that is resistant to antibiotics. This will give us insight. It's also used in, cancer, to be able to understand what actually is causing, what the genes that are responsible, or the biomarkers that are responsible for that condition in that patient. This help us to be able to gain insight, and help us for that to also manage the treatment of the patient. And it's also useful in forensic science. Of course, you must have heard of forensic science. It's basically using biology to solve crime or to investigate crime. So you notice that, in your room, you have your DNA strewn all around. Your hair could have fallen off one point in time, your saliva, your sweat. So when a crime occurs, it's possible that, the culprit could have left his or her DNA. Maybe during combat, there could have been an exchange of blood. There could have been an exchange of, um, what's it called now? Sweat. And, that could actually be taken as a sample, and then maybe we have a list of suspected individuals, and then the sample would also be collected from them and could be matched with the sample that was collected in the crime scene. So genomics has helped us to be able to solve a lot of problems, across different field of study, infectious disease, agriculture, personalized medicine, and forensic science, and in even other field of study like biotechnology. All right. So now let's talk about genome sequencing technologies. So we've been talking about genomics, DNA, understanding all these things. Now, how do we get the DNA? How do we get the DNA of an individual? How do we get the DNA of a plant, of human, to be able to understand or to be able to study it? This is where genome sequencing technologies comes in. So this encompasses the techniques that are used to sequence and analyze the whole DNA of an organism. So in the past, we've had traditional way of sequencing, the Sanger sequencing. But now we have the next generation sequencing, which help us to perform high throughput sequencing technique. You are able to sequence billions of DNA fragments at a go. And then you also have whole genome sequencing, which helps to determine the complete DNA sequence of an organism, and it provides the detailed blueprint of all genes and regulatory elements. So you can also sequence exome. Exome are protein coding regions of the genome. So we also have targeted sequencing, which is very cost effective, which helps you to be able to, sequence targeted region of, the genome. So this is in a case where you want to study a particular region that has been, you know, identified as um, identify as a disease-causing variant, or let's say you want to carry out evolutionary study. So, for example, we have some regions in the DNA of a bacteria, for example, 16S, that are actually used for identification and they are used for evolutionary study. And then we have 18S for fungi, which is used for evolutionary study. So this region can be targeted. Let's say you have a sample you're working with and you just want to carry out evolutionary study. So this region could be targeted and could be sequenced, to carry out your study. And then we have genome editing technologies, which is a very interesting technology which is used to alter DNA sequences. We have the CRISPR, which they also refer to as the molecular scissors. So which is clustered regularly interspace palindromics repeat. So it is used to edit a genetic defect or to correct a genetic defect. So let's say a sickle cell patient, a child who has been diagnosed with sickle cell. Genome editing technologies could be used to edit, such um, child child's gene, and in that case, it could actually correct that defect. Okay, so right now we're going to be talking about types of genomic data. So this module is actually to introduce you to, you know, the scope of genomic data, sorry, the scope of genomics, just for you to understand what genomics is all about, what you're going to be dealing with if you are going to be embarking on genomic research. And it's also to lay foundation to um, the world of genomics and bioinformatics. So we're going to talk about DNA sequence data. So these are some of the data you're going to be working with if you're finding yourself in the field of genomics. We have raw sequence nucleotide sequences, which is the ATCG of an organism. So it could be plant, it could be animals, just like I said, human, microorganism. And you have the gene expression data. This gene expression data is the information obtained from measuring messenger RNA transcription levels of protein coding genes in a cell. So this could help you to, identify the genes that are expressed in, let's say, cancer cells. Okay, and then we also have proteomic data, we have single cell genomic data. Now, these data are somewhat interwoven. Protein content information of a cell or that of an organism, that's for proteomic data. We have single cell genomic data, which is when you're trying to just study a single cell, you want to try and understand the information, the molecular information, like gene expression in an individual cell, let's say an hepatic, cell, someone that has liver disease, you want to study a single cell to understand the gene that are expressed in this condition. Maybe it's liver condition, liver disease condition, or cancer, liver cancer, lung cancer, or what have you. So these are some of the types of genomic data that we have. This is just a limited example. We have other types of genomic data. We have bulk, RNA-seq, and the likes. So this is just a few of the ones have highlighted here, a few of what we have, which I've highlighted here. Okay, so now we are going to move on to the next module, which is Introduction to Bioinformatics. I hope you've been able to gain one or two things in this particular first module, which is Introduction to Genomics. And I hope that I will also see you in the next module. Thank you. Bye for now. In this session, I'll be taking you through Introduction to Bioinformatics. In the previous session, we've been able to understand what genomics is all about. Now, in this session, I'll be talking to you about bioinformatics, what bioinformatics is all about. So here I have on my outline the definition and scope of bioinformatics, common bioinformatics databases and internet tools, data retrieval and bioinformatics file format, and DNA sequence analysis. Let's get started. So when you think about bioinformatics, what should come to your mind is that bioinformatics is actually an interdisciplinary study or an interdisciplinary field of study where computer science, mathematics, statistics combine together to generate, store, access, and analyze data and information relating to molecular biology. That's DNA, RNA, and protein sequences. Now, when you think about bioinformatics again, you think about it as a field of study that helps to make sense of genomic data, that helps to make sense of molecular data. So, when you look at your DNA sequences, the ATCG, and you want to understand the information that is encoded in it. I mean, for your ATCG, the ATCG is not just going to scream out and say, "Oh, I'm the one causing cancer," or "I'm the one, conferring antimicrobial resistance." So this is how bioinformatics actually play a role here to, make sense, to help us gain insight into this information beyond just, looking at the letters. All right, so I'm going to also talk about scope of bioinformatics. The scope of bioinformatics is actually very broad and it is rapidly evolving. Bioinformatics is that kind of study that doesn't sit still. So you have new research, new aspect of bioinformatics, emerging every now and then. So here I have genome analysis, we have protein structure and function, drug discovery and development, evolutionary studies, and database management. Of course, before the era of bioinformatics, biological or what's it called? Biological experiments were actually done in vivo and in vitro. But at the onset or at the advent of bioinformatics, we can now, carry out biological experiments on the computer. And that is why we have something like database where you could, refer to anytime you want to work with a particular data, be it genomic data, be it DNA sequences, protein sequences, or whatever sequence you want to, work with. All right, so we have common bioinformatics databases and internet tools. We have a lot of them. These are just the common ones that we have. We have NCBI, which is the National Center for Biotechnology Information. We have EMBL, we have Assembly, we have Uniport, we have ENA, European Nucleotide Archive, and then we also have internet tools, bacteria, viral, bioinformatics resource center. We have Use Galaxy, we have CloseVis. So CloseVis is actually used for visualizing the result. After you've analyzed your data, your genomic data, um, you can visualize it. This visualization will actually help you to have a glance, a summary of your results, which will actually be interpreted. And this visualization are often referred to as what you refer to as your figures, in your manuscript. So we have the NCBI Blast also. Now, there's something you need to know, which is quite interesting, is the fact that some of these databases also serve as internet tools. So, for example, NCBI could also serve as an internet tool. So that's why you see the NCBI Blast, which you can use it can be used to identify a sequence. So let's say, for example, you isolated a particular sample and you've done the DNA extraction, you've done the sequencing. You may have a proud knowledge of what the sequence is or the sample is, but you still want to carry out molecular study, and then molecular identification is part of what you have to do. So NCBI Blast is what you actually help you to be able to identify the kind of sample you're working with. If it's that you're working with a microorganism, for example, so that way you'll be able to identify, oh, if this is a Staphylococcus aureus, if this is an Escherichia coli, or if it is a Bacillus. And then we have the ANI calculator, which is often used when you are carrying out evolutionary study. Helps you to check for the similarity and the diversity of the what's it called, the sequences you're comparing. So let's say you have 20 sequences you want to compare. What I mean by sequences now is genomic data. Let's say you have different strains of a particular microorganism. This works well for when you're analyzing um, microorganisms. You can use this ANI to calculate how related, how similar they are, you know, based on their percentage, the nucleotide percentage, or how the nucleotide of this particular sequence is, related to the nucleotide of this other particular sequence. All right, so this is all for databases and internet tools. We have other internet tools that you can use, and the reason why they are called internet tools is because you can't use them without the internet. So your internet has to be on before you can use these tools, and of course, likewise for the databases. So these databases actually store different genomic data. Remember that in the previous session, we talked about types of genomic data. We talked about DNA sequences. We talked about single cell. We talked about what have you. You know, we talked about those kind of genomic data, and this database they actually created for specific types of genomic data. So your NCBI, for example, harbors different kinds of data. You have human data also. I mean, there's an integral part of NCBI which is called the GOE that you could use to, search for data that is related to humans. And then, of course, for the UniProt, we have data that has to do with protein sequences is stored there. In fact, we have a particular database that is actually designed mainly for pepper. So you will see something like pepper genomes. So as you're watching this video, you could just pause this video and then go on your browser, just type pepper genome. You would see a database that is actually created for pepper genomes. That is, if you want to do any study on pepper, you could just look that up. All right. So now let's talk about bioinformatics five formats. And this is quite interesting because the question you would ask yourself is how does DNA that is in a liquid form, how does it become, a file or a digital thing that we could now, analyze on the computer? So, and of course, you understand that this has to go to different process, from isolating your sample, from doing the DNA extraction, which is done in the wet lab. Now, during this sequencing, that is when these files are being generated into this sequence, the ATCG. So when these data are being generated, they could be stored in different file formats. And again, you need to understand that during analysis, or during, when you're performing analysis and you want to store the analysis, the result of the analysis, there's a way you also store them in different file formats. So it's like from one file format to another file format. So here it says bioinformatics file format is the standardized structure for storing and organizing biological data in a digital file. So you have your FASTA, which is a very common, file format in bioinformatics. So it's a text-based format used to represent nucleotide or protein sequences. Each sequence is represented by a single line description followed by the sequence data. And as you can see, that is what I have on my screen here. This is just a single description, which is showing you the sequence CCTAA, the ATCG, just like you have here. So think of the bioinformatics format as your PDF, your MP4, your PDF or Word document. So the reason why we have this thing is because remember we had talked about internet tools. There are some analysis that you want to do that require specific file format. So if you use the wrong file format, you're not going to get the results you're supposed to get. So that is why we have specific file format that is expected to be used in when you want to carry out an analysis. So we have file format, we have FASTQ. So FASTQ is used to represent nucleotide sequence and corresponding quality scores. And you don't need to worry. We're going to carry out an hands-on training on some of these internet tools going forward. And in fact, in this session, I'm also going to walk you through some hands-on as we proceed. All right, so we have FASTQ, which is used to represent the nucleotide sequence and corresponding quality scores. So quality scores, something you need to understand. Quality score is actually the score that is assigned for each nucleotide base as it's being generated out of the sequencing machine. So this is what the sequencing machine used to measure the confidence of each nucleotide base. So there is actually a standard. So when you have like quality score of 20 upward, it's actually good to go. But when you have something below 20, it means that the quality of that nucleotide base that is represented in that region is not actually what's it called now? Is not actually, accurate. There could be, some errors. So now FASTQ actually comes with this quality score. But this quality score will not come as a number, just like I said, 20, 30, 10, 20. So it comes in what we refer to as ASCII code. So this ASCII code is actually used to represent this number. So instead of having your 20, because imagine you having C to be represented as 20, is going to be clustered. So this at ERR, or what you're seeing here, sorry, what you're seeing here, they are actually ASCII code that is used to represent those numbers. So your 30 could be at, or it could be any symbol. So that those ASCII code are just those symbols that you use to represent the confidence of each nucleotide base. So that is what makes FASTQ to be different from FASTA. Why FASTA is just a single line description, but this comes with corresponding quality scores. And then we have the gene bank format. The gene bank format includes the information about features such as genes, coding regions, and other biological annotation. Just this is just a screenshot of what I have here is a bit more than this, but this is just a part of it. This is what a gene bank format look like. And this also, it can be used for analysis. So there are some analysis that you want to do. You know that the kind of file format that you need to get is gene bank format. So for example, let's say you want to carry out a particular analysis and you went ahead to use a faster format. Okay, for that kind of analysis, you're not going to get the expected result. So every analysis requires specific types of, kind of file format in that sense. So I have a last one here, which is okay, I think second to the last, which is GFF. So this is actually genomic fixtures, information about location, structure, attributes of various fixtures within a genome, such as genes, exons. Now, exons are, we have exons and introns. Exons are coding region. Because remember, when you talk about the genome is actually containing all the entire DNA sets and including the coding and non-coding regions. So the exons are the coding regions, while the introns are the non-coding regions, um, which are spliced during the process of genetic information. Yeah. So we have regulatory elements, and as you can see here in my screen, you will see that this is what a GFF file format looks like. And the last one I have here is BAM. BAM. So BAM is a compressed machine readable binary version of a sequence alignment map SAM file used in genomics to store aligned sequence read against a reference genome. So now, in the case where you want to identify variation, you want to identify mutation, and you're working on a particular sample, it could be human, it could be, plant, it could be microorganism, and you want to identify what variation has occurred that is making such, isolate, or what sample you're working with now to exhibit whatever trait is exhibiting. So you kind of map it to a reference genome, because the reference genome now is like the standard, or it's like the normal sequence that you have. So by mapping the sample you're working with that, you are able to identify what changes has occurred, and you can, yeah, what changes has occurred. Now, when you do that, when you map your sample to the reference, genome, the file format comes in BAM. Now, there is no much difference between BAM and SAM. BAM is just the machine readable binary version of sequence alignment. So SAM is just the same thing as BAM. BAM is just a compressed machine readable binary version of SAM. So that's just that. So this is the result. Remember, I said something about the fact that whenever you analyze a particular um, genomic data, you could also store it in a file format, or it comes out in a file format. So this is that part where you analyze a particular genomic data, and then it comes out in a file format. So for example, if you map a genome to a reference genome, it's going to come out in a file format, which is BAM. So all right, so now let's talk about DNA sequence analysis. And in this particular session, this is where we are going to really, do some hands-on, where you're going to see everything we've talked about. Because now you will see that without bioinformatics, genomics doesn't really make sense, because bioinformatics is actually what help us to make sense of our data, beyond just extracting the DNA, sequencing the DNA, and just like I said, when you have your sequences, it's not just going to pop out. For example, I mean, this sequence here is not just telling me that, oh, this is the region causing cancer, or this is the region conferring antimicrobial resistance, or it's just going to scream out and just say, oh, I'm the one causing cancer, I'm the one causing antimicrobial resistance. But bioinformatics is what actually now make us be able to gain insight into those letters, those language of life, to be able to get meaning to what they are actually doing. Okay, so here we're going to talk about DNA sequence analysis. It says here that it's the genetic sequence analysis which encompasses a range of techniques and methods used to investigate DNA sequences. So you've extracted your DNA, you've done library preparation, you've done the sequences, and just like you see, it came out in different formats. But now you now need to make sense of it, and this is where DNA sequence analysis, comes in. All right, so in this hands-on, what we are going to be doing is we're going to retrieve data from NCBI, create a folder on Geneious software, import sequence into Geneious software, call complementary strands, identify codons, also translate our sequence into proteins, also identify protein coding regions, open reading frame, and then I'm also going to teach you how to um, carry out primer, how to design a primer. All right, so now we're going to move to our browser. So you're going to go to your browser and search for NCBI. All right, so on your browser, you're going to type NCBI, NCBI, okay? So you're going to type this on your browser and you're going to click on enter. This will lead you to the web page. Just like I said, you have some databases serving as internet tools. So this was the BLAST, basic local alignment search tool I mentioned in the previous, session. So you're going to click on this and this will lead you to the NCBI database. Now on NCBI database, you have all databases. This is showing you different data sets where you could get your data from. You have Bio Project here, which is actually going to, which has like different project that has been done on different, genomic data. And you have Bio Samples here, you have some other data set here. So now we want to retrieve a data from NCBI because we want to make use of this data to show you. I want to make use of this data to show you how to perform DNA sequence analysis. You just want to analyze DNA, the DNA sequence, because it is one thing to extract your DNA, sequence the DNA, is another thing to be able to make sense of it, to be able to analyze it, to be able to draw insight from uh, such sequence. All right, so now we're going to make use of a bacteria. Just type Staphylococcus. So I can say 16X R. All right, so this is what I'm going to be making use of. But in order to streamline my data of interest, I'll just click on this drop down icon and then look for nucleotide, because the kind of data I'm looking for is a DNA sequence data, which is, the nucleotide, your ATCG. So I'm just going to look around for where we have nucleotide. Okay, so this is nucleotide here. Just click on nucleotide and click on search. Now, this will give you thousands of results, but you have to, you know, filter the kind of data you want to collect. So now we have, Staphylococcus aureus rRNA 16S partial CDS here. So we're just going to be getting one or two. So I'm going to be getting one. The other one is going to be just to show you what a gene bank format look like. All right. So now I could make use of this. Okay. This is Iraq here. You can see this is a sample collected from Iraq. So I'm going to check this box, and then to download, I'm going to go to this send to, click on this drop down icon, and then click on file. Now, this is where it makes sense. The bioinformatics file format I talked about, we have different file formats. So here you can see we have GenBank, we have GenBank Full, FASTA, and some other file formats. So we're going to be getting FASTA here because for the kind of analysis we want to do, we actually need to get a FASTA format. Okay, so there are some kind of analysis that you want to do that you will need the gene bank, but in this case, we are making use of the FASTA format, and you will also see what the FASTA file format looks like, even though I've shown you in the previous slide. Okay, so I'm going to click on this and create file, click on create file to download your file. So once that is downloaded, you just need to go to your downloads and rename, maybe rename as staff 16s RNA, whatever you want to rename it as. Now, I'm going to download this same sample as a gene bank format. So I'll just go through that same route and then change this to gene bank, and then click on create file. So now that we've downloaded this file, I'm going to go ahead to rename this file and put it in a particular folder. So it's going to be easy for me to upload it on Geneious software when it's time for me to upload it. All right, so go ahead now and rename. All right, so I'm going to rename this to staff. Uh, just to be able to differentiate it from this other one. Okay. So you can see this is what a FASTA file looks like. Just a single line description showing you the name, the genus and the species, and then where it was gotten from, the accession number, and of course the nucleotides. So now we're going to rename the other one STAB also, but in this case, gene bank. Can you just make it GB? All right, so we do not need to use this. We are not making use of this. I just want to show you how a gene bank file format looks like. So there you go. So you can see that this contains more details compared to the FASTA format. This is showing you a full details, some details that are not shown on, you know, the FASTA format. So the next thing we want to do now is to upload this particular sequence into Geneious software. So now, because I don't want to have my sequence lost in, haystack of some of the files I have here. So I'm going to create a folder to save this file so that I can be able to quickly identify it. All right. So I'm just going to do that right now. So you can do that also if you're practicing this. All right. So now that you've saved your file or you've renamed the file, I think it's just best to rename the file. Okay. If you can't open a new folder, just rename it. The next thing you're going to do is to open your Geneious software. And this is Geneious software. This is where you create your folder. These are some, you know, tools here that you can use to analyze your data. And then we have some, menu bar also here that you can also use to carry out some analysis. So what you want to do now is to create a folder on Geneious software. This folder is going to be created to have your file saved inside or to have your file imported into it. So you're going to click on this local here and then go to file, then click on new folder. So I'm just going to save this as GOGeneneBio. Once you create your folder, you're going to see your folder shown here and is highlighted in bold. And you're going to double click on this. Once you double click on this, a workspace will be opened. And here you have your alignment view. So whatever you upload is going to be shown here. And then whatever analysis you're doing, you're going to have them viewed here. Okay, so the next thing you want to do now to import the file you've downloaded from NCBI is to go back to that file again, this file here, and then go to import and then click on from file. This will definitely lead you to where you have your file saved. So this is under downloads, and I know that my file is somewhere here. Okay. So this was what I was trying to say the other time. I mean, you don't want to be looking for your samples in a stack of, a lot of files here. So I'm just going to search. Should be close. Should be close. All right, here it is. So I'm going to click on import. So now I've imported my file. You can as well go on to rename this from the accession number to maybe staff So just click on edit and rename it, but I'm not going to do that. You can do that at your own leisure time, just for practice. So now, like I said, once you click on this um, sequence that you've imported into Geneious software, you can see you have your alignment view or sequence view rather here. And if you increase, you would see um, your sequence. The nucleotide base is ATA. You can see like that in a linear form rather. So now you also could have it wrapped. So I guess this may be the first thing you will see when you import your data on your Geneious software. So you can simply just go to this, side of Geneious software. You will see wrap. So just click on wrap to make it, or check the box to make it linear. And then you can, zoom in and zoom out. All right. And then you could also expand this particular sequence view to make it broader, and you can, return it back to the, the former size. Okay. So right now, what we want to do is to, you know, identify the protein coding regions. Okay. Or better still, let's say we want to call the complementary, base, complementary sequence. But before you do that, I want to show you something. I want to show you an interesting thing here. So let's go to this percentage icon here. So if I click on this, this will actually show me some interesting thing. So you will see the length of this particular sequence, which is 1424, and the same is what we have here. And this is adenine. Adenine is the frequency of adenine. I mean, the number of time adenine was, represented in this sequence, if that is actually correct. So is 387. Then cytosine is 318. That is the number of times cytosine showed up in this sequence. That's guanine, and we have 409. We have thymine showing 310, and the percentage all over 100. So 27.2, 22.3, 28.7, and like that. And then you have the GC. Now, GC is always important, anytime you're accessing your sequence, because GC tells you the stability of your sequence. So when the GC is 50%, 60%, it shows that that particular sequence or sample is stable. And then the number of times GC content, or we have guanine cytosine bond rather, is 727, and we have 51.1%, which means that this is stable. And then the temperature that is needed to break this bond is, 85.42 degrees Celsius to break this strand. And you have the stranded DNA to be this weight, and then the double stranded. This is just like half of this. These are just the information that I just wanted to show you. Okay, so the next thing we want to do is to actually call the complementary sequence. So to call the complementary sequence, all you need to do is go to this bar chart icon here. Just click on this, and you would see some options. Okay, so before we do that, we can just let's try and yeah. So sorry, not meant to be this, meant to be this TV-like icon. So you can see you have complements. So you can just check this box. When you check this box, it will display the complement. Remember, we said that A bond with T, T bonds with A. I'm going to check that box again. Okay. All right. So, and we can change the color. Let me just change the color. Okay, by going to home, just go to home and change the color. All right, so that this will be more better. So now for the complementary, just click on this TV-like icon or display, and then check this box. Then you will see A to T, A to T, A to T, C to G. So this is the first thing you want to do. You want to check for the complement of this particular DNA sequence. Now, the next thing we want to do is to, translate this particular complement, translate it into protein sequence. Remember we said that the three letter, which is a codon, codes for specific amino acid. Now we want to see those things here. So by clicking on this translation here, okay, let's just uncheck this box, then click on this. So by clicking on this, you will see that we have ATA coding for isoleucine, CAT coding for histidine, GCA coding for alanine. And if you go on like that, you will see what we have. And in case you're wondering, when you see a star like this or an asterisk, it means a stop codon. This is a stop codon. That is, this is where the coding stop at. Now you may be wondering, how do I get to know this, amino acid? Once you place your cursor under this, you just need to check this tiny bar here, and you will see it's displaying those amino acids. So by placing my cursor here, you can see isoleucine. By placing my cursor here, you can see histidine. All right. So now that you've been able to identify this, to translate this nucleotide into protein, just like we talk about in the genetic flow of information, that is DNA replicates itself and it makes a copy of itself and into the RNA, and then the RNA translates those information into amino acid or into proteins, and this is what I'm actually showing you here. So the next thing now we want to do is to identify the protein coding regions. So you want to identify the protein coding regions. So you're just going to uncheck this box again. And then to do that, we can go to this chart, this bar chart, like what's it called? Icon or graph. And then click on this protein coding prediction. Just check this box. Once you check this box, you would see protein coding prediction calculating. And this is actually showing you, you know, the regions that codes for a particular, function. So now we can go back and, click on wrap. Just go back to home to wrap it. Just make it to make a lot of sense. So this is just showing you protein coding regions. You can see these are protein coding regions, protein coding regions. All right. So the next thing we want to do now is to check for those regions, to annotate those regions, and to do that, I'm just going to uncheck this again. I just prefer the Nelia form. All right, so to do that, just go to this particular arrowhead icon here and then click on it. And once you click on it, you're going to click on find ORF. Remember, this is part of what we also want to do. We want to find the ORF. So by checking this box, you will see that being displayed. And you can also click on this. And after you click on this, then you click on apply once you see this being loaded up. After this has been loaded, then you click on apply. Okay, I just need to hold on till that loads up. Right, so I'm just going to click on apply. Okay. Okay, just click on, sorry, I'm supposed to click on this. Okay, check this. Click on apply here. All right, so now if you scroll forward, you would see that you have, I mean, it's still almost the same thing though. If you click on this and click on apply, it's still the same thing. So the protein coding regions or find ORF is, those regions you could either make use of this or make use of this. Okay. Um, any of those works. So now you will discover that you have these regions that this bar, the orange like regions, this bar are all, protein coding regions. That is the code for specific, you know, protein. So you can see here you have proximal target signal, and if you place your cursor on that, you will see ORF interval, gives you the interval, gives you some more information there. Okay, so these are regions that code for specific function within the sequence. While this is actually a 16S RNA sequence, yet not every part of the sequence codes for specific functions. And that is why we, check these ORFs to be able to identify those regions that codes, okay? Those regions that codes, and this is what we have. So the last thing we're going to do here is to actually design a primer. This is quite interesting. So primer is, you know, a short sequence that you actually design is between 18 to 22 base pair or sometimes 24, as the case may be, which you design in order to for you to use it to, amplify a specific or targeted region in whatever sequence you're working with or whatever sample you're working with. So you design your primer to amplify it. So you sort of use it as a template in your PCR to amplify. So for example, let's say you are looking forward to amplify a particular region in the sequence you're working with, then what you want to do first is to design the primer. So now let's take this sequence as the sequence we want to use to design our primer, and we already identify, the region we want to design the primer from. So what we need to do is go to this icon here. You can see primers. Click on this and then click on design new primers. All right, so we have different options here. And here you will see that you have forward primer and reverse primer. Of course, when you want to do your amplification on PCR, you need your forward and reverse primer. So you're going to check this. And then here you will see you have generic, standard primer, precise. This design primers to amplify specific regions suitable for cloning. So this is suitable for cloning. We'll just leave this at default. And we could also include the region. Now, let's say you already know the specific region you want to, you know, design your primary from, or you want to get your primary from, then you can include. But we're just going to leave all this at default, and then we click on OK. So now the primer is being done. So now we have 10 results found. So if you come here, you would see these are my primers. So we have 5 because it is both forward and reverse. That is why it is 10. So let me just amplify this. So I can just decide to scroll. These are my primer. These are my primer. Let me just make this a bit, all right. So these are my power. This is forward and this is reverse. 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 Forward and reverse. So now whenever you want to get your primer, you must ensure that both the forward and reverse falls under a coding region. So for example, you see that these actually all fall under, should we say under a coding region? That may not be, I may not be able to figure that out now. I think it fall under this a little bit. So, but this is just in a case where you need to really design a primer. I mean, this is just for explanation sake to just show you how to design a primer. So once you do that, what you want to do is to extract the forward and the reverse of the primer you design. So let's say you want to get one out of this. Let's say you want to make use of um, let's say this first one. So let's just make use of this one. So you're going to right click on it and then you click on extract regions. Just click on extract regions and OK. So now you have the first um, forward of the primer, that particular primer has been extracted. So the next thing you want to do is to extract the reverse. So to extract the reverse, just come here also and right click. And as you can see, you will see the temperature. This is the normal temperature that you need, for your primer, 60, the length 20, just like I said, between 80 to 22 or 24. Then you also right click on this and click on extract regions. Okay. So the next thing you want to do after you've designed your primer, which is the forward and reverse, is to import them out of Geneious software and then send it to the lab for them to identify, to amplify this specific region. Okay, so and then what you need to do next is to right click on this. Sorry, you just need to check this box and go back to file. Okay, and then click on export. Export. Just export, batch export. So now, when this window pops up, you want to change this file format to faster. You're not going to leave it at Geneious format because if you leave it at Geneious format, by the time you want to open this file, most likely it will open Geneious software for you. Okay, so what you want to do is click on this drop down icon and change it to faster sequence, not faster compressed sequence, but faster sequences slash alignment. So you're going to click on this and then you click on OK. So definitely this is going to be on your desktop and click on OK. So, and also click on OK. So after you're done doing this, you can then forward this primer you've designed, both the forward and reverse to the lab for you to be able to identify to amplify what specific region you want to do. So this is how you design your primer, and this is just what I'm going to show you on how to analyze your DNA sequence by, calling the complementary strand, identifying the protein coding regions, the ORF, and then also translating your, DNA sequence just to make sense of what you've been talking about, you know, especially when we're explaining the genetic flow of, you know, information from the DNA to protein. So right now, I'll go on and access what I have downloaded. All right, so on my desktop here, you will see my primer design. So I'm going to open this. This is forward. You can see this is very short sequence. This is the forward, and this is also the reverse. So this is what you're going to send to the lab, when you want to amplify a particular region. All right, so that will be all for this DNA sequence analysis, and I will see you in the next module 3, where we're going to be working with whole genome data, identifying genes that confers certain traits. All right, so I'll see you in the next. Bye for now. Welcome to the last session of this aspect of Introduction to Genomics and Bioinformatics. In this session, we are going to be going through a more practical approach, and I believe that you're going to have a real world applications of genomic data analysis. I believe that you've also been able to learn some things in the previous session that we had, and I'm sure that in this next session, you would see how these things connect all together. All right, so let's get started. I have in my outline here data acquisition and preprocessing. Now we've done some of this, a bit of this, not some, a bit of this in the previous session where I showed you how to collect data. But in this particular session, I'm going to be showing you more more aspects or more things you need to do after you've collected your data, the likes of data processing. That's when you want to access your data, if it is of good quality or if it is of bad quality. Matter of fact, this particular, approach is applicable to any kind of data you're working with, even if you're working with an RNA-seq, you know, you also need to check for the quality of your data. So in this session also, we're going to be anno performing functional annotation. So functional annotation is basically identifying what function the genes within the genome they perform, and significant function or functional annotations are the antimicrobial resistance, the virulence factor. Yeah, this particular session is actually you know highlighting microorganism majorly in in another aspect, which is going to be taken by another facilitator. I think a transcriptomist, human genome will actually be worked on. So this is just this particular session is going to be highlighting uh, microorganisms. Okay, so we're also going to be performing comparative genomics and evolutionary study. This will help us to also perform multiple sequence alignment and then construct phylogenetic tree. So we'll be considering two types of data. We'll be considering a partial CDS, and also we'll be considering a whole genome for this analysis. So here is what we have under data acquisition and processing: retrieval of raw sequencing reads from NCBI, SRA, quality control and trimming using tools like FastQC and TrimGallo, featuring removing low quality species and adapters, genome assembly combining short reads into longer content. Now, the thing about data preprocessing is it is different for it has some nuances. Okay, there are some data that you have to perform quality control first before you move to you know genome assembly. Now that's if you're also using an internet tool. There's some internet tool that you could singly perform what's it called, quality control. But there are some internet tools that once you perform genome assembly, um, quality control is part of what is integrated into the genome assembly tool. So quality control trimming will be performed automatically once you submit your data for genome assembly. So I'm going to explain this as we go on in the practical, in the hands-on. All right, so under functional annotation of genes, we'll identify antimicrobial resistance, the patterns, how some genes confer antimicrobial resistance and how they go about, this mechanism. Then we're also going to check for virulence factor. This is actually to help in tracking outbreaks, to understand or to gain insight, to have an evidence-based insight on resistant patterns in pathogens or, you know, pathogens basically. All right. So, and the last one I have here is comparative genomics and evolutionary phylogenetics or evolutionary study. So this is study of how organisms evolve and adapt over time by comparing their genetic makeup. So, for example, you realize that and this has actually been used in identifying new outbreaks, new strains, for example, COVID, from Omicron, now we have different strains of COVID that has, you know, broken out, emerged over time, and that's due to evolutionary study. And scientists bioinformaticians were able to identify this as a result of evolutionary study, as a result of genomics, being able to see that the genetic makeup of the initial strain is different from the new outbreak by comparing the two together. So this is what phylogenetic analysis does. By comparing two strains, three strains, four strains of a particular genus of an organism, it will help us to understand the variation that has occurred over time, why this particular organism is behaving this way and this other one is behaving that way. So this is just basically what it is in the simple form. So we're going to be comparing genomes across species or strains, studying evolutionary relationship, using multiple sequence alignment, identifying conserved and variable regions, and building phylogenetic trees to infer common ancestry using Geneious software or Mega. All right. So now we're going to get started by first, collecting our data of interest. All right. So now that you are on NCBI, we are going to be getting some sequence on NCBI. So we're going to be getting a partial sequence and a whole genome because we can actually perform phylogenetic, we can build phylogenetic tree with whole genome, and we can also build phylogenetic tree with partial CDS. I mean, just like the 16S RNA I downloaded from NCBI in the previous session, you can use that to actually build phylogenetic tree. Remember, I mentioned that this particular kind of data is used to, for identification, and it's also used, to construct evolutionary study to study evolution, of different species of organism. All right, so now we're going to look for some number of what's it called species. Let's still work with staff. So staff, and then let's streamline this to equal side. So what we want to do is basically, you know, um, get most different strains of staff, the 16S. So if you are asking me why we're using the 16S, you know now that the 16S is actually used for evolutionary study. But this doesn't mean that you can't use a whole genome for evolutionary study. But it's just going to be um, you may not have to limit your study to evolutionary study because there are other things you can do when you have a whole genome. I mean, you can, just like we're going to do in this video, we're going to identify antimicrobial resistant genes, virulence factor genes, and of course, if uh, we are able to also check for mobile genetic elements also. All right, so here we have Staph aureus 16S, and then we'll click on search just to get more other sequence. Now here we are okay. I think I may have to complete. Okay, I'm just going to then leave this to all databases. All right. Um, Apologies. That should be 16s. So imagine just because I didn't, had 16s, knows why I had that. So you want to be sure you are inputting the right keywords also. This is very important when you're searching for, you know, this is very, very important when you're searching for, your data of interest. So now I'm just going to click on this. Okay, so we're going to get about 15 more apart from the one we got yesterday. I think this was the one we got in our previous session. So I'm going to just randomly select other ones I have here. Okay, just going to select this, going to select this. That's three. Still go down a bit. And if you want to check for the number of, you know, samples you've selected or data you've selected, you can simply, look it up here, selected four. So we have about 10 more to go, 10 or 11 more to go. So this doesn't have anything to do with, having to select a particular data. I'm just trying to select a random sequence here. So, okay, going to pick this. I won't pick this. This is too short in comparison to the kind of data I want to use. So you want to be on the lookout on the kind of base pair, sequence base pair you want to also select. So now we have eight. Let's just pick let's pick this. Let's pick this and this. Okay. So these are just staff, different species of staff. We have Staph aureus, we have Staph sp, we have Staph felis, Staphylococcus bonari. So these are just different species, and you can decide to make use of different strains of the same species. You can decide to make use of different genus. It depends on what you want to do. Like this is in terms of research now, genomic research. Depends on what you're, and you can decide to also limit it to geographical location, or you just want to get sample from, a particular geographical location. And if you're going to do that, you just need to type the name of the geographical location you want to get the sequence from. And for you to be able to confirm that, you may have to double click on this. Let's say this is the data you want to, some of the results. So you have to click on it and then scroll down to check for the location. This is where you're going to check the location. Okay, this data is actually from Iraq, as you can see. So let's say you just want to work with, data from Iraq. So that's how you check by accessing or clicking on the link of the specific data. So, okay, I think we have eight already. So let's select some. Okay, I think we're good now. So the next thing we're going to do is to come to send to and, you know, click on file, then create file. Sorry, we're going to change this to faster. Don't forget, change it faster. The file format you're getting is faster. If you get a gene bank and you want to construct phylogenetic tree, it's not going to work. This is where, you know, file format is actually very important. So you just click on this and then create file. So automatically that will be downloaded. So you just need to go to your download and rename that particular file. So I'm just going to rename this to rename. And just make this staff and others. Right, so this was the data we collected in the previous session. I'm just going to be performing phylogenetic tree, constructing phylogenetic tree with this. Okay, so now we're going to go to Geneious software to import those files. All right, so now that we've opened Geneious software, what we're going to do now is to import our file into Geneious software, just the way we did when we wanted to, analyze the sequence. By the way, I would like to also say that you can annotate your data on Geneious software, and that was what I was trying to show yesterday. Yeah, so show annotation. So annotation is different from finding ORF. I just need to clarify that. So when you annotate, it's actually going to show you, the specific function of that gene, just like you see proximal target signal. So this is the function. Annotation is basically identifying the function of the gene. So when you hear the word functional annotation or annotation, is basically you identifying what function does that gene perform, or what function does the gene, yeah, what function is it carrying out, within the genome. Okay, so that's just to clarify that. So now that we've opened Geneious software, we'll go to local, just like the procedure we took when we import our file. So this is just the normal procedure. You click on file and then you import from file. So definitely your file is going to be under downloads, and if you have it on desktop, you can also check your desktop. Oftentimes you may need to, have your data well, you know, well organized, so you don't have to be searching for your data just the way I'm doing now. So this is what I'm looking for. I'm just going to click on Control select and Control select. So those are the two files I'm going to be importing. Just click on import. So now there's a question that you'll be asked immediately you click on import. Now you will see that here it says keep sequences separate, create sequence list. So anytime you see these two options, what you should click on is keep sequences separate. Remember that we downloaded those sequences together, except for the first one we use for sequence analysis. So in this case, we have to keep them separate because they are separate sequences, but we just downloaded them together on NCBI. So you're going to click on this, keep sequence separate. And as you can see, Geneious software has helped you to keep the sequence separate. Now, there's something you also need to do, but I will not be doing it in this video because of the duration. If I'll be doing that, that might take a longer time. So there's something you also need to do anytime you import your file on Geneious software or you import your sequences on Geneious software. You need to rename. Under name, you can see that you have this under name. So you just need to rename this by right clicking on it and edit. You can see edit name. So you can give it the name of the strain. So you can just say strain R1. Just copy this. You can just copy this. Just click. You just need to click on this. Okay. Just need to click on this and then copy this strain and then use it to rename this. Okay. So I'm just going to leave mine this way, but it's good that you rename yours this way so that by the time your phylogenetic tree is built, you can be able to also trace those names. All right, so you can see these are some of the information that we have: the sequence length, the GC content, and um, the topology. It is linear. So now that we have this sequence here, what we want to do is we want to construct phylogenetic tree. And you can construct phylogenetic tree without having multiple sequences. That's why this is called comparative study. It's a comparative study. So you would highlight every of this sequence and then you will now click on this align/assemble. Because the first step you need to take or the first step you need to perform before constructing phylogenetic tree is performing multiple sequence alignment. So you're going to click on this align/assemble and you will see here you have multiple alignment. It's multiple alignment because you are performing alignment of more than two sequences. So you're going to click on this. After you click on this, you will see different options you can use to perform multiple sequence. So multiple sequence alignment has different methods. We have the Geneious alignment method, we have muscle alignment method, we have plus W, and the rest of others. So either of these works you can decide to use, but most of the time people use Geneious alignment and muscle alignment. And the reason why you should take note of the alignment you use is in the in the coming days, if you have a research you want to carry out and you have to construct phylogenetic tree, it is important that you document the method that you use under your materials and method. Okay, so in this case, I'm just going to leave it in Geneious alignment and then I'll click on OK. So another difference between this Geneious alignment and muscle alignment is that I think the algorithm is just what makes them different, and the fact that one is faster than the other. So you can test that in your own time when you want to perform multiple sequence alignment. You can decide to use Geneious alignment for one, and then you can decide to use um what's it called, muscle alignment for the other. So there's something that we forgot to do in this, what's it called, in this session. We forgot to actually create a folder. So while I was trying to import, I didn't create a folder. So my folder is actually inside local. You understand? So, but let's just leave it that way. And I can decide to create new folder again and just give it file tree. All right, and I can decide to move all this cut and then bring it here. All right, so easy peasy. So now that we've done sequence alignment, you will see that we have nucleotide alignment here. This has aligned all the sequence that we have all together here. So you just click on this nucleotide alignment, and you can see your sequence view or alignment view. In this case, you can decide to zoom in. So now let's zoom in and let's access this. You will discover that you would see some similarity across this, sequence. You will see this is let me try and expand this for all the sequence that we actually align together. Now this is supposed to give me a name, but let me try and see if I can tweak that a bit. Okay, so this is showing me their name. So you can just come here to show, instead of show names, just, you know, put it at description. So because the names are actually under the description. So now you have the name. So you can see for this particular, we do not have anything here. So these are gaps. These are gaps. These are all gaps. These are gaps. And then we have guanine, showing you that in this region, they share the same nucleotide base. They share the same nucleotide base. And if you also check for that, you would see there are some regions that are actually highlighted. And you will see that in this region, we have A almost all true, showing that there's a level of similarity here. And you have this TT here. Now, this color TTT and CCC are the conserved region. They are the conserved region. That is across that particular strain, they are conserved and they do not just change. And then we have some places where you have, what's it called? Variable regions. You can see G, A, you can see T, G here. Some variables are here. While you have some regions that are conserved across these specific strains of Staphylococcus aureus. They are conserved. They are conserved. So these are just basically what multiple sequence alignment helps you to be able to gain insight to. And in this way, you already know, how similar and how divergent these sequences are. But you may not really be able to get full access to this until you now construct the phylogenetic tree. So right now we're going to move on to construct the phylogenetic tree. So now on your sequence alignment, you're going to go to this icon, this tree icon, and then click on tree to build your phylogenetic tree. Now every of these methods stated here, genetic distance model, tree build method, and all these things, they are actually important for documentation, just like I mentioned when you want to carry out, your multiple sequence alignment, you know, and you need to document the specific method you use for multiple sequence alignment. So these things are also meant to be documented in the case where you're carrying out a research and need to build multiple sequence alignment, and you need to build phylogenetic tree. Okay, so now you're going to leave everything at default except this, you know. So you leave this at bootstrap, then you change this to 1000, 1000, 1000 to 100. So this is just um, a standard way of, setting the random seed. So that's the number of times the phylogenetic tree is performed, and then it chose the best fit, the best results rather in the process. So now this is out group, and this out group, this is when you're trying to compare different strains of a particular organism together, and then you now want to check their similarity or if they are closely related to maybe a suspected outgroup that you feel like they share the same, what's it called, the same trait together or the same characteristics together. So let's say you're comparing Staphylococcus aureus and you want an out and you have an outgroup, let's say Escherichia coli that you want to compare it with. Escherichia coli is an outgroup in this case because it's of different genus. So in that case, you have to identify your outgroup within the sequence that you actually uploaded. You understand? So this is how you're going to go about it. All right. So this is how you go about this. So now that you've set all these parameters, what you need to do next is to click on OK. And after you click on OK, automatically your phylogenetic tree will be constructed. So basically, this is how you construct phylogenetic tree. You can see what you have here. You will see your tree here. You can see um, this what these are what we call the nodes. These are the nodes and the root nodes. So you can see that we have this was what I was talking about. Let me try and collapse this. This was what I was talking about when I was mentioning you renaming your tree. You know, when you don't rename it, most likely you're going to have or just the accession number. So, but when you rename it, most definitely you're going to have the name shown here, and that way to help you to be able to compare your tree together. So if you check this tree, you'll discover that you have some values here, and let me just quickly explain this. Now we have different branches. We have, for example, let's start from the low point here. You have these two sequence coming from this recent, the same recent ancestral root, and they have 99% bootstrap value, showing that they are closely related. Now, if you compare these two trees together with these at this root, you discover they also share 78, percentage of bootstrap value in common. But by the time it's at this point, this actually branch, you discover that there's a long distance between this particular sequence and this sequence. So this value is just showing you the similarity and the dissimilarity of this sequence across, the branches. So now we have this, we have this, we have this, we have this. So we have this branch, we have this branch, and we have this branch, we have this branch. So we have some clusters also here. This is like a cluster, a whole cluster here. This is another cluster here. So, and this, just like I said, this value is showing you how related they are. And if you check the multiple sequence elements that we perform, you discover that there were some similarity. There were some similarities actually, from the multiple sequence alignments. You can see that they are having the same nucleotide basis across the region. So that's why you'll see that there was not really much, you know, um, what's it called? There's not really much dissimilarity, just a few. This and this also. So now the fact that these guys belong to the same ancestral root doesn't mean that they have to have the same, they have to have, how do I put it now? They have to have a high percentage similarity. It could mean that why they evolved from recent ancestral roots, a lot of, what's it called? Adaptation or evolution must have taken place between both of them. So that's why you will see that uh, at these roots, it is 59 percentage. Okay. All right. So now let's move on to the alignment view. Okay. This is the alignment view also. I mean, this is just the same like as what's it called? The multiple sequence alignment result. Then we have the distances. Now, there's something interesting about this distance. This distance, or, or let me call it heat map, is actually going to show you the similarity by comparing, all the sequence together, comparative. That's why it is called comparative study. Anytime you're doing evolutionary study, it's comparative study. So if you compare this strain with this strain, of course, you're comparing it with itself. That's why you see that it is very dark and it's not giving you a value. But if you are comparing it with this particular strain, which is here, you will see that it has 97.3 percentage, and it's kind of a bit, arch in color. So when you see these different shades of color is actually showing you how closely related they are compared to this compared to some ones that are, quite, white. So you can see that even with that, it's still 95.8 percentage. So that's to tell you that all these, strains are actually very closely related, very, very closely related. In some cases, this white is just showing you the highest dissimilarity that we could have. And this is not actually dissimilarity. I mean, if you have a 95.8 percentage value, that's to show you that they are actually very related. And you can also just change this to numbers only. This will show you, an overview of what's it called now? An overview of the comparison, the similarity of these sequences. So you can see that they are closely related. They are very closely related. Okay. So this is also what you would do when you're calculating ANI, average nucleotide identity. This is what it also looks like. Okay, so now let's go back to uh, Geneious software workspace. So now, after you've constructed the phylogenetic tree, you can decide to, you know, um, download this tree. All you just need to do is to go to File and then save as image file. Just save as image file, save as image file, and you can adjust this depending on how you want it to look on your document. So you can also change this to whatever format you want to change it to. So I will leave my at PNG, and then I will leave it at but and then click on OK. So just click on the file name, file go. Okay. So after I've done that, I can decide to access it by going to my downloads, and then I can open it. So here it is. So if you're not satisfied with this tree, you can decide to, adjust it during the time you're saving it. All right. So this is how you perform phylogenetic tree, you know, on Geneious software. So now you can also perform phylogenetic tree for whole genome. Remember that the sequence you actually worked with here is actually a partial CDS. You can also do this on, you know, on or with on whole genome, yes, WGS, whole genome sequence. So I'm going to be showing you how to do that. But before I do that, I'm going to show you how you can also collect a whole genome sequence on BVBRC, how to perform quality control, and you know, and other things I've listed on the slide. So now we're going to proceed to the next thing by making use of a particular, what's it called now? A particular internet tools, which is BVBRC. BVBRC is one of the internet tools I listed in the previous session. So now we're going to proceed to our browser and type BVBRC. So just go to your browser and type BVBRC. So BVBRC is actually a resource center, bioinformatics resource center, where you can use to analyze different, choose different sequence rather. So this is what you're going to be using in the rest of this session. I'm also going to make use of GUse Galaxy though, just to show you how to perform um, quality control. So I'm going to put a pause to it in this session, and in the next session, we'll continue from this point. Thank you.
This session, we're going to be looking into functional annotation of genes. Under functional annotation of genes, we're going to be identifying antimicrobial resistant genes. We're also going to identify genes that confers virulence factor. This is essential to help us to be able to track the resistant pattern of what data of interest we are working with or what pathogen we are working with. So now we're going to proceed to our browser to type NCBI to get the data of interest. This was the same thing we did in our previous session when we are trying to work with 16S RNA for phylogenetic tree. So now we're just going to type Bacillus subtilis. It could be any other data of interest. And now, whenever you want to work on, any data of interest, especially when you want to perform functional annotation or you want to perform a broad analysis, it is essential that you work with a whole genome. And that was why the previous session was different from this session in the sense that in the previous session, we just carried out, evolutionary study by constructing phylogenetic tree, and we made use of a particular marker or biomarker for that, which is 16S, which has been known for identification and evolutionary study. But in this case, we want to carry out a broader analysis, identifying antimicrobial resistance and also identify genes that confers virulence factor. So it is essential that we use a complete sequence or a whole genome sequence. So now in our data set here, we're going to filter to SRA. SRA. So SRA is sequence read archive. And the reason why we are making use of SRA is because SRA harbors data that are raw directly from the sequencing platform. It is yet to be pre-processed. So that is why we're making use of this. And because I also want to show you how to perform genome assembly from the start. Now, why we had said that we're going to perform quality control in the previous session, I would like to say that we will not be doing that in this session because transcriptomics actually covers that. And again, because the platform we are using, which is BVBLC, quality control is an integral part of it. When you submit your data for genome assembly, quality control is an integral part of that, such that if the data is bad, there's going to be some trimming and all. So that is why I'm not going to cover that in this session. But if you want to know how quality control is being performed, you can refer to the transcriptomics session. All right, so we're going to click on this. And then you can type WGS here, search. And then there's going to be hundreds or thousands of results. So you have to ensure you're getting the right data. So in this case, we are looking for whole genome sequence. And most often we have this data appearing in, sizes like this. And if it is a metagenomic sample, for example, it could be much more than this. You could see them in gigabytes, 1 gig, 2 gig. So now, when you want to retrieve this data, you don't have to necessarily download in this case. All you just need to do is to copy the raw ascension number of this data. So for example, let's say I want to work with this particular data. Let me scroll down a bit. Let me just make use of this. So let me click on this whole genome sequencing of Bacillus subtilis. So now you see we have some information here. If I click on this, you see this will give us more information where this, data is actually cutting from. We have some information here showing us geographical location, collection date, host it was collected from, and let's just go back. So now this is what I was talking about, the run ascension, so which is SRR35, and like so. This is actually more like an identifier that you use to you can submit this. You don't necessarily have to, because in a case when you're working with a larger data, you don't want to be, let's say you want to work with 10 samples. You don't want to download all the 10 samples, like I mean 1 gig or 2 gig. That will be 20 for 2 gig. So in this case, what you want to do is just copy this and have it, documented in an Excel sheet, and proceed from there. So right now we're going to copy this particular sequence, copy the run ascension rather, and then we're going to go to BVBRC. So because this is a bacteria, yeah, you could perform quality control on your bacteria sample on Use Galaxy. The same process you use for an RNA-seq, which will be covered in transcriptomics, is also the same way you're going to perform your, your quality control when it comes to bacteria. But I mean, you don't have to go through all that trouble when you know that when you submit your data on BBBLC for genome assembly, your um, automatically quality control will be performed. So right now, what we want to do is to submit this sample for genome assembly. So what's the workflow? The workflow first, before you can gain insight into your data, you have to be able to perform genome assembly. Okay, because this is a raw sequence, and genome assembly help us to piece together these, pieces of reads so that we can be able to, carry out genome annotation. Now, this is being said in case when you have a sample you're working with, or let's say you have your own data you want to work with, and the raw sequence is being sent to you, and then you're asking yourself, how could I make sense of this data? So the first thing you have to do is to carry out genome assembly. So now to carry out genome assembly, you're going to click on Tools and Services. And under Tools and Services, you can see genomics here. We have genome assembly, genome annotation. So you're going to click on genome annotation. So now we have different options. After you click on genome annotation, this web page is going to open. We have different options with which you can submit your data for genome assembly. Okay, so you have paired with library, you have single read library, and then you have the SRA run assertion. And this is the route we actually use. So paired with the library is when you have a sample that comes in forward and reverse. So the forward version of that sample will be uploaded here, the reverse will be uploaded here, and if it is a single read library, it's also going to be uploaded here by just clicking on this drop down icon, and or if you already have it uploaded in BVBRC, or you go to this folder icon and then you go to this arrowhead icon also and click on select file. Then you can locate the sample in your computer anywhere you saved it. Okay, but since that's not the route we are making use, and the same works for single read also. But in this case, we're making use of SR run ascension. So you just need to copy and paste that run ascension from NCBI. And then you click on this arrow icon to validate this particular data. Now, remember I said something in our earliest session. I talked about the fact that some of these databases, they share data in common. They share information in common. So for example, let's go to ENA. ENA, that's European Nucleotide Archive. So if you click on this particular link, and then you come to this session here, enter session, if I paste that particular run ascension and click on view, it's going to generate some information. You can see Bacillus subtilis, paired, library paired, and some other information. And I could also download here, but in this case, we are not downloading because we don't want to waste data. All right, and in the case where you have enough data, of course, you can download. All right, so you're going to click on this arrowhead to validate. After it has popped up here, it means that this particular data is valid. And then the next thing is to choose the parameters you want to use for genome assembly. So if you click on this drop-down icon, you have different assembly method that you could use. You have unicycler, you have Spadis, you have Canon, other methods. But in this case, you're going to leave it at auto. So BVBRC will be the one to choose the assembly method that best fits for this genome assembly. So the next thing we want to do is to create an output folder. So to create an output folder, you're going to click on this folder icon here and then come to this cross you're going to click on this icon that looks like first aid and just click on create folder and then write GOGeneneBio. I'm just going to make my GOGeneneBio practice. Click on create folder. After you created folder, the folder will be listed here. So you can scroll down and just look for that particular folder you just put there. So here it is. So you're going to click on this and highlight it and click on OK. So after you've done that, the next thing is the output name. So you can make use of this and as the output name, you can just Bacillus subtilis underscore underscore and copy the SRV and then click on assemble. So after you've clicked on assemble data, the assemble data will be generated, which you will then download. So I'm going to show you how to do that. So once you submit that, you just need to go to workspace and click on my jobs, and then it will lead you here. Remember, after we perform genome assembly, you just go back here and then see. Sorry, you're going to click on my jobs. Then you see your work here running. So in order not to waste time, this may take a little bit of time. So after the genome assembly has been performed, so I'm just going to make use of one of the samples I've worked on here. So now you can see we have this. So let's use this as an example. This is genome assembly. So we have other results here. Let me check. So this, you don't have to be on your computer while this is running. I mean, this could be done in the back end. So you could just close up your computer and then come back to it to check. Okay, so this take about 15-20 minutes. So you just go back. Let's just go to the results we have here. So when your assembly is done, you just click on this. You will see a result like this, assembly report. By the time you click on this assembly report, all right, so once you click on this particular folder, this is the folder I'm actually talking about. But before we click on this, let's just check this assembly report. So for this assembly report, you're going to see information, show you how the reads are actually being pieced together. So this is just like a bandage plot showing us description of that. Then you can see that the assembler that was used was unicycler. the assembler sequence, the time that the assembly was done, the command line, this is like a back-end work. Then some other information we have here, so there's something I want to show you here. So you can see here you have good context file. This is just to tell you that genome assembly also performs, quality control. And you can see trim. This is actually showing that the reads that are bad have been trimmed off. Okay, so that's why we have this result here. So now let's just go back to this. So this is what you're going to download. So this is in context.faster. So the idea is whenever you perform genome assembly, your results, short reads being pieced together will, result into context. So that's why you have something like context.faster. So you can download this particular file, and this is the file you're going to be using for genome annotation. Okay, so after you've performed genome assembly, the next thing to do is to perform genome annotation. So genome annotation is what gives you insight into, or what helps to make sense of your data, to be able to get insight into what the data is all about. The genes that confers antimicrobial resistance, the genes that, the resistant patterns, not just, the antibiotics they are resistant to, but how they go about the mechanism of resistance. Okay, so now we're going to download this file. Okay, so by just highlighting it and then come to this green bar side and then click on download. So once you click on that, the next thing you need to do is go back to tools and services, and then you will see genome annotation. Now let me let you in on something. So here is comprehensive genome analysis. For comprehensive genome analysis, the difference is that when you submit your sample under comprehensive genome analysis, it will automatically perform genome assembly and genome annotation. But the reason why I had to show you all through the process is so that you can understand the workflow whenever you want to analyze your data. And in some cases, you may not be using BVBLs. You may have to use other internet tools that will afford you to use them separately. So that is why I have to show you this. So now that we've downloaded our data, we'll go to genome annotation, click on genome annotation. And then here you're going to upload. You can see it says parameters context. So that is why we had to download that particular, file format, context.faster. So in this case, you can't just upload any kind of data here. This is where bioinformatics file formats now make sense, knowing that you can't just upload any kind of data in this particular. So as you can see, the service has set a faster formatted content file. Can you see? So you can upload a FASTQ format here and it will work. It will generate an error notification. So in this case, to submit your data, all you just need to do is go to this folder icon and then go to this arrowhead icon, click on it, click on select. So to locate that data that you just downloaded, so you just go to download, and this is the data we just downloaded. So click on this and click on open. So you will see your data has been uploaded here. The next thing you're going to do is to click on start upload. Start upload. So after you've done that, you are going to then select the recipe, the annotation recipe, by clicking on this drop-down icon. So bacteria, if you're working with viruses, you put viruses. If you're working with bacteriophages, you put that. But in this case, we're working with bacteria. So we're going to click bacteria, and then we're also going to click on the taxonomy. So let's just type this. You have to type here Bacillus So Bacillus subtilis. So here you're working with, let's do that again. So you're working with species. So let's just scroll. So you click on species and then the identifier. So your identifier could be Bacillus or the strain name of this particular sample. And we can as well copy the SRA. Just so after we've done that, automatically the output name will also be here. So the next thing we're going to do is now, by the way, this particular naming, I mean, my label is not a one size fits all. I mean, you could decide to name your sample whatever you want to name it. It's just necessary that you don't get yourself confused whenever you're naming your data. I mean, that's in the case where you're working with multiple sequences or multiple data. So now our output folder, we can just click on this drop-down icon and make use of GOGeneNeBio practice as our output folder. So the one we created initially when we wanted to perform genome assembly. So we're going to click on this. And after we've done that, this particular tab, Annotate tab, will be highlighted automatically. So all you just need to do is click on Annotate. Now, after you've clicked on Annotate, you can access your results by clicking on this job list or make use of the workspaces route by clicking on this and click on My Jobs. So either of them works. So let's make use of this since we already shown you how to make use of this. So we're going to click on this job list and you will see that we have this already. So what we want to do now, we can see that the genome assembly we uploaded is still running. So this might take a few minutes. That was why I didn't use this particular data anymore because, I can't be waiting in this session. All right, so we're going to click on refresh to refresh it so that we can see the job we've submitted. So sometimes when you submit a job, you may see that it's not popping up here. So all you just need to do is refresh the page. And this will pop up. So, and interestingly, these have already been performed. So let's just click on this and see what we have. So still the same thing, context.faster, the assembly report, just like I shared, in the other sample that I was using to teach. Okay, so now that we've done genome annotation too may take some time. So I'm going to make use of a sample I've already uploaded here. So let's just go back and see some of the samples we have here. Okay, so we have Bacillus subtilis here. So I'm going to double click on this. This is what your annotation result is going to look like. As you can see up here, annotation. So let's scroll down. Click on this. And then you will see different results here. But here, to access your result, you need to click on this eye icon. So once you click on this eye icon, this will, lead you to a world of this particular packaging. This is where you can explore different aspects. But in this particular, session, we are limiting this to identifying antimicrobial resistant genes and various factor genes. So now, for you to be able to access that result, you can see that we have different tabs here. So to access it, you're going to click on this specialty genes. Specialty genes, by the time I place my cursor on this specialty genes, you will see specialty genes tab contains a list of specialty genes, e.g. antimicrobial resistant genes, virulence factor, drug targets, and human homologs. So since my focus is on antimicrobial resistant genes and virulence factor, so this is my go-to tab. I'm going to click on these specialty genes first. After I've clicked on that, then I need to filter, because when I place my cursor on specialty genes, there are different genes confirming different traits under this tab. So I need to filter it down to antimicrobial resistance. So to do that, I'm going to click on this funnel-like icon. I'll click on this, and then this will pop up. You can see I have all those properties here. So antibiotic resistance, drug target, transporter reference factor. So we have 56 results for antibiotic resistance. We have 6 results for virulence factor. So now we're going to access antibiotic resistance first by simply clicking on this. And after you click on this, you're going to watch out for this table here. So in this table, you have interesting results. And for you to be able to identify these results clearly, you need to do some filtering by coming to this tiny cross icon here, bordered with circle. So just click on this. And then what you're looking out for is antibiotics, the antibiotic class they're resistant to, classification, which is also the same thing as the resistant patterns. Okay, the genes, of course. So these are the basic results that you collect whenever you want to analyze your data for antimicrobial resistance. So you can see we have genes here. We have the LIAR gene, EF-gene, G gene, which is resistant to peptide antibiotics. So anything peptide antibiotics, this particular gene is resistant to it. So in the case where the patient is administered peptide antibiotics, the pathogen, will develop resistance to it. And you can see if you scroll down, you will see other genes. And again, you see the antibiotic class and then the antibiotics, specific antibiotics, daptomycin, fusidic acid, okay? So, and then you have streptomycin and other antibiotics. And again, not just that, you also get to see the resistant mechanism. How do these genes, how do they, perform? How do they go about their resistant pattern? You see regulator modulating expression of antibiotic resistance. If you see something like efflux pump, you see efflux pump. That is when the bacteria pumps out the antibiotics out of itself, reducing the concentration within itself. That way, the antibiotics will not be useful. So you can see different resistant patterns. So these are the results you're going to get from antibiotic resistance, basically when you're performing analysis for antimicrobial resistance. And in the case where you have your data to work with in the nearest future and you want to check for antimicrobial resistance. So, and you can simply just check this box, okay, and copy. Right, so when you check this box, you just copy and then you could go to your Excel sheet and just paste, okay? Sometimes this may happen. Just go back to BVBLC and you can just check this box. Check this again. I don't know why that occurs sometimes, but I mean, it works most of the time. So just paste it. And then you can see that you have your result here. So this is gene classification, antibiotics, and antibiotics class, and antibiotics. So these are the genes. And for this space that are not filled up, you can just delete them. Okay. So just delete this. And this is also not classified. So you can delete those. I mean, this is more like data cleaning in a sense. So now let's go back to BVBRC to check for virulence factor. So now that we've checked antibiotic resistance, now we're going to uncheck this, click and then click on virulence factor. So you'll see you have genes that confers virulence factor here. So you can see adherence, biofilm formation, intracellular survival, and replication. Because in this case, you don't need antibiotic class and antibiotics. Basically, what you need is just the gene and then the classification. So you also copy this and then paste it on an Excel sheet, maybe a separate Excel sheet. That's for the research case when you're trying to analyze it, some data. All right, so this is how you perform virulence factor. I hope you've been able to gain something in this particular session. If you have any question during the live session, we're going to take questions. So this is where we're going to take a pause to eat for this part of GOGeneNeBio. I hope you've learned something. And I hope you've been able to, understand how to make sense of data. Peradventure, you come across any data in the nearest future. All right. Thank you. Bye for now.